So let's start part 3, which are the applications of GeneMANIA, how to use GeneMANIA to answer different types of query. So we know that we can have two types of function prediction and two types of question. So the first question is, what does my gene do? We have one gene as a query. And the second question is, give me more genes like this. We have a gene list as a query. So first example, query one gene, and it's APOL4. So here we are going to use the GeneMANIA web app because it's a rapid query. And the aim here is that, okay, I got APOL4 for my project. I need to work on APOL4, but I've never worked on it, so I have no idea about the function. So I could read a lot of papers, but before doing that, what I'm going to do is a quick GeneMANIA search to understand the function of the gene. So I enter APOL4 into GeneMANIA, and then I look at the resulting function, which is pore complex and nuclear pore. So then I look at PubMed, and definitely APOL4 is an importing that helps importing some proteins into the nucleus. So then we got a good guess from GeneMANIA here, and it saved me a lot of times because I didn't have to read all these papers. Then second examples. I have two genes here. Give me more genes like this. I'm going also to use the GeneMANIA web app because it's just two genes and it's also a rapid query. So I knew that these two genes, SUS12 and EZH1, were part of the polycomb repressive complex 2, PRC2, but also knew that they were not the only two elements of this complex. So I wanted to retrieve in a rapid way the other elements of this complex. So I enter SUS2 and EZH1 into GeneMANIA, and I have here the resulting networks in the middle of the page that gives me all the genes related to these two genes. And then if you compare to the image on the right that display the component of the PRC2, then you can see that GeneMANIA had incorporated these elements into the new added genes. Here, a third example. So now we have 23 genes. So give me more genes like this. And these 23 genes were coming from an Alzheimer's disease research story. And these 23 genes, they have a mutation. So they are known to, or they were found mutated in Alzheimer's disease. But very often when we have these genes mutated in different patients, it's very difficult to find results if we do like a simple pathway enrichment analysis. So my guess here is that maybe I'm not going to have global function, but if I do a simple pathway enrichment on these 23 genes, because it's not enough to give me some common function, but if I use GeneMANIA, I may see how these genes are connected to each other, and I'm going to expand the network, and then I may be able to predict the function. So indeed, then you can see here the physical interaction in the network, or we'll see here's some genetic interaction. And so the genes with the black border are the query genes, and the other ones are the ones that were added by GeneMANIA. So we can see direct interaction between the genes of the query, but also we can see some more final networks that is has some function, for example, a neuron part and neuron projections that seem to be very well expected for early onset of Alzheimer's disease. So GeneMANIA help us to expand the network and to find some functions. Another example here of the question, give me more genes like this. So we start with 43 genes, and here we have used the Cytoscape app. So this is based on a paper from the Gary Bader's lab, and the way why they used a Cytoscape app is because they they add multiple layers of information. So we have here the resulting GeneMANIA network based a lot of shared protein domain based on the color of the network. And but they also added the node color, and the node color represented expression data for these genes, and the red showed a upregulation of these genes, blue a downregulation of these genes. And they also played with the node center and the node border to display the two time points that they had. That was 3 hours and 28 hours. So this is an example that in Cytoscape you also can use the GeneMANIA as a base network and then add on top of it multiple layers of information. So, and then in this example, then the query has 502 genes. So we use the Cytoscape app because when you have more than 100 genes, it's better to use the Cytoscape app, and the other link to the web version may be too slow if it's working. And here, actually, this is to show you the link between enrichment map and GeneMANIA. So when you have an enrichment map where the nodes are the pathway, you can right-click on the pathway and of interest, select apps and select enrichment map, and then show in GeneMANIA, and it will create a GeneMANIA network of the genes in the pathway. And this pathway was NC RNA metabolic process was a large pathway with 500 genes. And actually we used GeneMANIA and the function in GeneMANIA to subdivide this large pathway into more precise pathways. Then, as a last example, as an advanced topic, so we use GeneMANIA with the default functional interaction networks, but you also have the possibility to upload your custom networks, and then you can use the GeneMANIA algorithm in the way you want. And here it's an example on how GeneMANIA algorithm has been built to create a patient classifier, and it's called NetDX. It has been developed by Schrader Pai, Shirley Yu in Gary Bader's lab. And then you see that they use exactly the same algorithm, but their functional networks were based on their own source of data. So now, as the last topic, we are going to speak over two related apps. And the first one is String. So String is a database and web resource that provides information on known and predicted protein-protein interaction. So you can add here in the search box your gene of interest, and you will get a network of the related genes. So it's going to be like direct interactions. And so it's not for gene function prediction. And this is the output. So based on the all genes in the network, String is also doing pathway enrichment analysis. So you also can download all the interaction data. And String has a web version but also is integrated in Cytoscape app. So this is the search box where you have String, and this is the same search box where you can also select GeneMANIA. So you can select GeneMANIA or String and get your String network. And also you can do the pathway enrichment analysis. So the difference between GeneMANIA and String, so String is protein focused, and it uses direct interaction to score nodes, whereas GeneMANIA is gene focused, uses thousands of networks, and uses label propagation to score the nodes. So, and another last tool to get functional interaction networks is FunCup, and it covers 21 model organisms. It's only a web version, and they have a large choice of very interesting functional networks, and it's very interesting to look at it as well. So this is ending this lecture on GeneMANIA. So we would like to acknowledge Craig Morris for his slide, and you can see this original lecture, and the GeneMANIA developer team, Christian, Max, Ruth, Quaid, and Gary Bader. So here are some reference. So the video from Quaid, the GeneMANIA documentation where you can have all the details as well as the GeneMANIA original paper. So it's time to get hands-on. So let's dive into two practical lab, one using the web version and one using the Cytoscape app.